So now we can move to trying to understand the functions of each organism group in a healthy food web. So how much available energy do various plants send to their roots? So I've mentioned, I don't know how many times, to push the point very hard, that each of the different plant groups are like in successional levels. So when we look at our food forest, we'll walk through creating a climax state in a forest by understanding the soils tied to the successional movement. And that's how we can get our food forest to go so fast, by working on the soils. So weeds, they take 20% of their energies fixed into their roots. What does that mean? 80% of their body is outside and only 20% they use for root growth. Thus, we don't find that they're very, what we might describe as nutritious, because they don't tend to take up as much nutrient. They don't have that much relationship. And we tend to find them in poor soils. So they developed over time to basically live in very poor soils, right? And they're not very based on nutrient, or what we would call our macronutrients or micronutrients that we see for health, what we need to, say, survive, our building blocks. Grasses, okay, so this next level, and look at how the difference, how fast they move into going up. So 60% of their energy they put into roots. So we'll see grasses, especially in the first level and second level grasses, or what second level being vegetables, how much they'll put into roots. So much more root structure, creating a much more what we describe as nutritious plant. Vegetables, okay, the classifications that we then would classify as vegetables. In this instance, 75% of their mass is in roots. So again, that takes up more and can create what we have as this nutrient dense, and we particularly like those foods more so than what a weed is, because obviously you can eat a weed. What's the difference? Maybe you could say it's bitter, but we have a lot of affiliation with eating bitter foods over another bitter food because of the affiliation of nutrient density, and that's tied to root mass. And trees. 80% of a tree's energy is for its root system, okay? And then then we can see how fruits or trees, nuts, have very dense nutrient density in their things because they have such a big root mass. So all of these have a relationship with the exudates that we were speaking about, the glue or polysaccharides. 50% of the energy from energy into roots is released. So the energy is taken through photosynthesis. 50% comes in to keep new plant growth, leaves and growth. And 50% is sent down to the roots. And when it gets to the roots, that's in the form of what's called simple sugars, proteins, and carbohydrates. Okay? So in a sense, you cannot distinguish nutritionally between those things. So if I was to take a Big Mac here and I broke it into on the back, right? Proteins, sugars, carbohydrates, salts, that's what it's made out of. Some of it could be processed. They might add in other chemicals to make it last longer. But basically, that's the same thing as a plant, okay? These building blocks of whatever we're eating. So glucose is normally what they're after, the bacteria. So why would plants release exudates, glucose and fructose? They want minerals. So that's that relationship created through photosynthesis to get exchanged to the bacteria. Bacteria is some type of relationship. Scientists don't know exactly yet what and how it works, but why. But I tend to believe, and it's not a provable thing, just, hey, it seems to make sense to me. I think common sense sometimes is what's missing in science. Look, the fungi eats the dead tree after it dies. So if it ate all the baby trees, there'd be no food left and it'd die. So it lets the trees grow up by feeding them, and then it eats it at the end. I mean, hey, come on, it's like what we do with chicken, right? Except for unless you're a vegetarian, then we eat them at an unfertilized embryonic stage. Yes, okay. So why would we want to feed these microbes? Well, that's us feeding them. Why would I go out of my way to feed microbes? Why don't I just let the tree do it? Well, if I can increase the microbial populations by feeding them, and if I understand what they like to eat, right? And if there's more of them to elicit, when they die, they're taking the minerals and their bodies bring the minerals into their bodies, and then when they die, they're water soluble. The tree can then take up the water soluble minerals. So how do I grow bacterial colonies? So when we do our compost tea, we'll take our compost, we're going to add it to a water solution, warm water, air, so air, water, darkness in a bucket, and we'll add in fructose, sugar. the bacteria will then breed, and then we'll add them to our soils to improve the bacterial relationship. Same thing happens if we take our worm tea. We'll take worm castings, and in worm tea we're after the enzymes. So we want to breed those enzymes. So same thing, we'll brew them with sugars to increase the microbial population, to add to our plants, and we basically feed the microbes to the plant. The plant ends up eating the dead microbes. So these would feed other organisms that benefit to the plant. So they die, the other organisms eat them, they die, the plant gets water-soluble minerals. So what are the benefits? Well, if we do this, when we feed what we call effective microorganisms, the good guys, because also I haven't spent much time on, and not going to go into too much detail, the non-effective microorganisms like algae and different types of bad bacteria, yeast, and things like this, right? So they have these things that also can live and proliferate. So if we see like rotting food and things, these are the bad guys. And when we see our compost pile, we don't see those because the good guys don't let the bad guys in. And those are, we'll do more in the mushroom course, but the acenomycin. So how am I going to facilitate the development of acenomycin? Acenomycin is the dominant bacterial mycorrhizal colony, and acenomycin only has one friend. Fungi. So he says you can be here, but all these other guys, you can't be here. So even if their spore drops, mold, drops onto the compost pile, it can't grow because acenomycin says you're not allowed here. But functionally, it kills it. So they can't colonize. So they consume the nutrients, the foods, and that might breed harmful pathogens. So therefore, we don't see these types of harmful pathogens in our soils, in our garden, if we facilitate the oxygen relationship, oxygen and water. So what are the benefits of these things? What are these actually when it breaks down? There's nitrates, and nitrites. These are what's going on. So these bacteria convert nitrogen from nitrite to something that does not enhance disease organism growth. So the disease organisms like to get nitrites. But our bacteria eat nitrites also and convert them to something that those organisms can't have. So if we help and support the good bacteria by feeding them, they outcompete the bad bacteria in the soil. Thus the bad bacteria stimulating problems with our plants like root diseases and fungal issues with our plants are developed by the bad ones. So we're going to feed the good ones, keep down the bad ones. So what are the benefits? Well, if we have the good guys there, because we're colonizing our good guys through composting, compost teas, right? And we're helping our soils, well, they're going to occupy the space. Yeah, so if I'm already here, you can't come. So I guess I shouldn't have put it up. So in here it's dirty, right? Air is dirty, it's full of mold, bacteria, spores, yeast, and for sure I'm dirty. Touch my face, rub my nose. Now that's got to be dirty, right? Well, if I open this, and now it's exposed, and I touch it, why doesn't it get contaminated? It has acenomycin, and the fungal network has already colonized it and said, I'm here, you can't come. So even if I put a spore in, it's 1 against 1,000,000. Well, 1 doesn't beat 1,000,000. Okay, so this one they're just now, right now, killing what I just put in. Get out.
So if you cut it open, it would kill it, right?
Depending on how much you opened it and then facilitated like stuff. So then what happens is those things want to eat too. And this is a food source. But that's why we don't really open it until this is colonized. So then the entire food source has been colonized. So even if it is open, like the one that's in the garden, remember the one that bed and the mushrooms are flushing? Because that food source that it's still deriving, right, is colonized completely by mycelium. So it can't get contaminated, right? So that's the issue. So we'll look at that more on Sunday. All right, so what this creates, acenomycin basically, and the facilitation of this through our things is disease suppression mechanisms. We want to suppress diseases by facilitating the good guys. So we use exuates so that no food's left over for pathogens. So in the garden, you don't have the diseases. Our plants are really, look healthy all the time because there's no space facilitated in our soil because of our integrated soil regime, and they can't facilitate. So you don't walk in the garden and see like mold and and yellow spores and all this weird stuff, right? Because there's no place for it. The good guys is saying, hey, you can't be here, mate. So other disease suppression mechanisms, things like penicillins or poisons. Actinomycin produces antibiotics or and inhibit compounds, toxins to prevent pathogen and pest growth. So then soils that are high in the good guys, which we call effective microorganisms, don't allow pathogens, and we don't allow pests. So like even the insect comes down, lays its egg, but the good guys eat the eggs. So then it breaks the cycle because he doesn't have a place to breed. Okay, so we can see that like in different types of insects that you might find. Other disease suppression mechanisms, if something does get hurt, maybe I went and got some ginger and I took off a section of the rhizome so I can have tea. But that means that that's now an open wound. It's like cutting off a piece, and I eat this part, but now it's a wound. And then I put the dirt back. Why I said that please put the dirt back. If you don't put the dirt back, then that dirt can't protect the wound, and then it'll get diseases because of the air. But if we cover it with the compost has antibiotics that will not allow that to get diseases, so root diseases. So if we do all of those things in our garden, right, we don't really have issues with disease protection. There's no more pesticides needed. I don't really have issues with bugs very much. And the little bugs that we have are controlled by their population through a natural balance. So I could look at it like this. I don't have to spray pesticides because I only lose 10 or maximum 20% of my yield. And I'm okay with losing 20% of my yield, understanding I don't have to have input, my time, money, or poisons in my garden. Okay? Next, I have nutrient retention. The nutrients, when I water, are not leached through my soils because I facilitate the exudates or polysaccharides that bind the nutrients into aggregates. So when the water goes through, the water comes out clear, and I don't leach, which is called leach it. I'm not leaching the nutrients into the subsoil or our aquifers. That includes carbon sequestration, and it stops leaching. And the nutrient availability, since it's there in the bacterial colony, when the plant wants it, it's in the right forms, so not nitrites, it's in nitrates to nitrogen, in the right forms at the right place at the right time when the plant wants it, okay? So then the plant is never starved of nutrition, it's not starved of water because of the water retention, okay? It doesn't have problems with pathogens, and all of this is just basically a function of building your soil food web up so that your plant lives in a healthy condition. And if a plant is born into and lives within a healthy condition, it's going to end up being a healthy plant. Also, if we do have toxin levels, and sometimes we have places in the world that have high lead contents and things like this, decomposition of toxins in our soil, which is called, we have two ways to do this, photoremediation and phytoremediation. So photoremediation would be if I grew a particular plant that liked to take up like lead, and then I removed the plant, the lead would go away, and then we could deal with that in another way, okay? Or if I add compost, then I can dilute the toxins in the soil, which is called phytoremediation. So I make compost and I dilute the toxin down to a manageable level because we measure toxicity by parts per million. So if we dilute it, the parts per million come down to what would be considered an acceptable level. Also, when the toxin comes through the bacteria, they bind it into what's called it's basically a stable form. So we have in all of our toxins what's called stable and unstable. What that means is certain things like plutonium and radiation things, in the unstable form, it will leach through the soil and go into the water table. Then when we get our water, we have these toxins in it. But if we can phytoremediation them, they'll bind up and they won't become water soluble, and they'll stay in the soil, but they don't go into the plant and they don't leach. They just sit there. Okay, so that's what they try to do with these remediation techniques, especially for places like toxic waste dumps and things like this. So bacteria and fungi don't wash away. If I take this and I water it, they don't leave. They're bound, right? And they're holding the soil in place. So that's why when we finish tomorrow, on Sunday, when we take the old ones out, though all the nutrition has been converted into the thing, this is real dense. I mean, you break it, they hold the soil together. So this is something that's also a benefit. So they need their food, they like it, and they stick to it. So they stick their bodies onto the food source. Bacteria glues are formed, right, at a pH of 7. And fungal threads, which are called organic acids, so they're described, it's an amino acids, if you will, create the what we know as amino acids are creating those fungal threads. And one of the major ones is called glutamine. And so that's what's binding up the soil, okay? And that's what they look like under a microscope if you're looking at those threads. So, cool. Bacteria and fungi, they're more concentrated in nitrogen than any other organism. So now we can start to look at different organisms and see their mineral profiles. So how much minerals are found in different organisms, starting with the bigger ones, and you can go all the way through the profile. So they retain nitrogen longer than any other groups, like for instance bacteria, fungi, people are made up of nitrogen also. Green leaves, obviously we can see protozoa, nematodes, deciduous trees, conifer trees. So you can start to measure the nitrogen retention in any of these plants and or animals and what nitrogen is cycling through the soil. So different of those groups like what's called a carbon-nitrogen ratio. And this is important if I want to grow a mushroom. I can't grow the mushroom if I don't get the carbon and nitrogen ratio right. So carbon is easy, but if I don't have the correct nitrogen, and that's why it was important when we did our compost pile. We were adding carbon and nitrogens, right, to try to get, and if we measure it properly, we can actually, or scientifically measure it in a laboratory, the actual carbon-nitrogen ratio. So bacteria like a 5-to-1, 5 times more carbon than nitrogen. Fungi likes 20-to-1. So if I want to get a good flush, I need 20-to-1. So we'll learn on Sunday how to adjust that. Green leaves, what we would describe as lettuce, things like our second successional grasses, they need 30-to-1. Protozoas are 30-to-1. Nematodes operate in 100-to-1. Okay, so then these are different regions. So these are things that we're going to look at and how we would, and why when we make compost, people are working on trying to maintain this carbon to nitrogen ratios. Okay, that's why it's really difficult. It's easy to put the carbon in, and you think the nitrogen is equal in volume to carbon, but it isn't. Because the nitrogen, right, the leaves, that's not still equal in volume. So that actually what they're after is the ratio they tell you to use, but they're looking at this ratio, which is different. But we'll more detail in the mushroom class on how to exactly balance your carbon and nitrogen ratios. So here's your nitrogen cycle, your plants and animals, plants growing, the animal eating the plant, defecating. The defecation goes through the soil, so this is called a denitrification bacteria. So we have different in the soils, kingdoms, if you will, or colonies. So the first one is called nitrogen fixating bacteria. So these bacteria grab the nitrogen and they fix it, and then they defecate, and it goes through to what is called free nitrates, nitrites, and ammonia. So when the ammonia comes out, so if we start to do compost at the beginning, you get more of ammonia, but towards the end, it stops that smell, because the other set of bacteria, which is called denitrifying bacteria, they start to convert the nitrates into nitrites. And then when they release that releases the nitrogen back to the atmosphere. And then the nitrogen can be taken back down by the plant and put back into the cycle. So this is, without this cycle, or we call geochemical cycles, that was the book Lovelock and Gaia theory. Without the understanding and the facilitation of our geochemical cycles, then life on Earth would die. So we can then look at these forms of nitrogen in our soil, and we want to say some of them are leachable, means that they'll leach out with watering into our subsoils, and then we can get high nitrates into our water. And this is a big problem with agriculture, where a lot of times they're NPK, they're putting in very leachable forms of nitrogen, and then that through erosion and their events can go into our river systems, or it will leach into the aquifer. And then a town that's nearby to this farm ends up having high nitrate soil, water, which is very ill health for us. So the different types of the most leachable forms, nitrates, nitrites, ammonia, any of these forms of nitrogen, they leach very poorly. They don't stay in the soil. The least leachable form is pure nitrogen. So we have to understand, how do we fix pure nitrogen into the soil? Well, we have to understand those two colonies of denitrification bacteria, right? And how we have the nitrification bacteria to the denitrification bacteria to the most non-leachable soil form, which is nitrogen. Now, what does that for us? Compost. Okay, so pretty simple. So least leachable bacteria all the way down to organic matter. So organic matter that we see is very leachable. That's why when we're watering, did y'all notice the leachate comes out and the color changes? Yeah? But once we get stabilized, that's at the beginning. Once we get stabilized compost, right? So it becomes more stabilized. Then we don't see as much leaching because the fungal networks are developing and they're grabbing it. And then the leaching, the color changes is not as prevalent as like dark brown. Okay, so that's why we want to focus on trying to not leach our soils. So nitrogen, so why is that important? Well, nitrogen in these organisms as part of the living organic materials such as proteins, amino acids, and amino sugars. So these are basically, once we get through the soils to get into building plants and animals, or functionally build plants and animals eating plants, or other animals, but either way, it's still based on plants. The plants create these amino acids, amino sugars, and proteins. So we want to then move into where they come from. How do we build them? So these substances resist microbial degradation and account for the slow availability of nitrogen in the soil. So when the microbial population dies off, so if I make compost tea, some of those microbes that I put will live, but the majority of them will die. So within the 1 to 3 days, that's why we see that, wow, the plant just responded, because it took those deaths of all that millions of microbes that we make, and then it gets that in a water-soluble form, and then the plant takes it up and it gets that boost. Think about it like organic Miracle-Gro. So they become available. So we'll do that with compost tea regimes. Then we'll start to look at when should we put compost tea. So it's not just apply it every day. We want to apply it at kind of the right times. So we'll apply it like when we get the plant started, and then we won't apply it during its growth cycle too much, and we'll apply it right at yielding. Okay? So we give it to it at the appropriate times. So vast amounts of carbon and nitrogen are held in the atmosphere. Okay? So we have this atmospheric carbon and nitrogen cycle, and they're cycled into the plants in various forms. But nitrogen can be in other forms, so we want to look at those. But nitrogen is important. It's the basis of the whole system, but maybe not the basis of health. So when we start talking about human health tied to nutrients, we don't typically describe nitrogen, though it makes the basis of plant growth. So what are these other nutrients? Where are their reserves in the soil? Where are these nutrients at? How do I work on them if they're important? And how are they held in the soil? How do they become plant available? So if the nutrient's there, but the plant can't take it up, it doesn't do any good. So we need to understand how to get it into a water-soluble or nutrient uptake form for plants. Plants can't take it up by themselves. And we need to understand soil structure, nutrient pools, and retention.